Hi, welcome to another section of agricultural biotechnology. In this section, we shall see some of the example of gene transfer techniques that has revolutionized agricultural biotechnology by taking the example of BT cotton, herbicide tolerant soybean, virus resistance papaya. Now let's look at this papaya. Would you feel like buying it? No, because you know that it has been spoiled or it has been infected. And yes, the answer is it has been infected by the papaya ring spot virus. It is a pathogenic plant virus belonging to the genus Potivirus or virus type P. This occurs in two strains, that is the P infects the both papaya and watermelon, whereas the type W infects only the watermelon. And these appear like a long rod-shaped particles, non-enveloped filamentous virion, which are 800 to 900 nanometers in length. They are single-stranded linear RNA. Look at this, the black one what we see, that is the genomic RNA, which is of 12 kb in total, and it is encapsulated by the coat protein, the green one what we are seeing. Now, this picture shows us this papaya is not ripened, but still you can see the symptoms very easily. So the unripe papaya are green in color. Whenever these are infected, we can easily identify whether this papaya is infected or not. Like such type of concentric circle or C-shaped structures you can see. Okay, so these will be darker than the background color. That is, if your papaya is green color, these whenever it is infected region, it will still appear in darker green color. And when this happen, it can affect the vigor of the fruit and trees and fruit quality. That is, it doesn't spare anything. It will affect the entire tree. For example, you can see that the yellowing and the vein clearing on the young leaves, yellow molting of the leaves, severe blistering and leaf distortion, dark green streaks and rings in the leaf stalks and stems. So these are the some of the symptoms which clearly shows that your papaya plant is being infected by papaya ring spot virus. And how usually it is spread? It is carried from plant to plant by aphids during the feeding. That is, aphids is the background picture what you're seeing. That is when this aphids feed on the infected plant, and then it goes and sits on the another healthy plant, and it feeds on that. Automatically the saliva, okay, of this aphids will be carrying the virus, and thus it spreads to the healthy plant. It is not spread by the other insects and does not survive in the soil or dead plant material. That is why we can see that if the entire farm is being infected by this particular virus, what they do is they either burn the entire field. Also, it's spread by the movement of infected papaya plants. It is not usually seed transmitted, but there is a case in Philippines wherein they have observed that the seed from which that papaya tree was formed that was infected, and it was reported that maybe the seed had carried the virus. So now how are we going to genetically use this gene transfer technology to establish a virus-free papaya plant? That is what we are going to see that. Now, this was a big breakthrough. Okay, the papaya ring spot virus was slowly working its way through the Hawaiian Islands. The Hawaiians' main cultivation was this papaya, and they came to know that all the papaya plants were being infected, and the traditional plant breeding had no solution. So the only solution was to burn off these plants, which led to the huge economic loss. So the scientists turned to a brand new technique that allowed them to move the genes from one species into another, creating new source of genetics that could introduce DNA from the virus into the papaya, and that it might work something like a vaccine, somehow protecting it from the virus. So we all know that genetically engineered organism is one that has the genetic material from the different species. So the scientists made use of this idea. So what they did was they identified the gene which coded the code protein. Okay, this is the gene. They identified it. And then what was their idea? To infect this gene into the papaya plant. So once what happens, it is acting like a vaccine. The papaya plant will integrate the gene, gets integrated into the papaya genome, and it also multiplies like any other genome of the papaya plant. So when the actual virus is infecting the papaya is ready to face it because it is like as though it is vaccinated, and it can defend itself from the papaya's ring spot virus. That was the entire idea behind it, because this technique had worked out in tobacco and tomato plants. So the same technique was used. The scientists used the vector which was carrying the DNA that contains the virus code protein. There was a reporter gene that acted like a marker gene to track where our DNA of interest is, and the promoter gene which expresses the gene of interest. And they packed all this, and then they wanted to transmit it into the papaya plant. Then they had a hurdle, because how will you going to insert this packed vector into your papaya plant, because there is a cell wall which is acting as a shield. You can't. Okay, the cell wall is acting as a shield. It will not allow the easy transfer of your vector. So then they use the modern technology, that is your gene gun, okay, wherein they coated the DNA with your gold or tungsten particle, and then they fired it into the papaya plant, which directly goes and sits into the nucleus of the papaya plant. Now that plant is being called as a modified, genetically modified or transformed plant. So now they want to check, really, did the transformed plant has that capacity to withstand the virus. So they did an experiment. So they choose the control plant and the transgenic plant, which is now having the gene for the food protein. Now they intentionally infected both the plants with the papaya ring spot virus. Obviously, as you all guess, the control plants will die because they are not resistant to that papaya virus. Transgenic plants, because they are being transformed, they have to survive. But most of them died except for one. Maybe this was because when we spoke about the gene gun technology, we told that when we are shooting or firing, some of the plant may be damaged, or the DNA will not enter into the plant. Maybe this was the reason. Okay, the possible reason is not known. So this one plant which survived, what they did was they took the cell, that is the explant of this plant, used to from this explant they developed the callus, and then they developed the shoot root and regenerated into the whole plant, and they cloned the plant and gave it to the farmers. And especially what they did was, there were two important papaya cultivars, okay, that is the sun up and rainbow variety. So the scientists successfully created two virus resistant varieties of papaya. This was the sun up and the rainbow, and both had the resistance against this papaya ring spot virus. That is how they use the growth protein mediated resistance to bring about the resistance against the papaya ring spot virus. And this actually worked really well in the Hawaiian country, and still they cultivate the papayas using this genetically modified plant. The another method is the RNA mediated resistance. The papaya ring spot virus contain non-cloning sequence called as sat-RNA. cDNA of the sat-RNA PRSV. PRSV means it is papaya ring spot virus. Okay, the cDNA of sat-RNA papaya virus is introduced into the papaya through the TI plasmid. So what it means is the papaya ring spot virus, it has the sequence, non-cloning sequence, which is called as sat-RNA. So what the scientists did was, since this is RNA, we wanted a DNA. So how we'll get a DNA from the RNA? I told you use reverse transcriptase. So you'll get a complementary DNA. So they constructed a complementary DNA of the sat RNA of the papaya virus, and then they introduced it into the papaya. So this was introduced into the papaya. This is the complementary DNA of this sat RNA which is present in the virus. So through the TI plasmid they introduce. Now you know what are TI plasmid, tumor inducing plasmid of your agrobacterium. So when they produced like this, papaya produced the viral sat RNA which decreases the virulency of the virus. So this was another method. So the papaya plant started producing the sat RNA of the virus, which indeed decrease the virulency of the virus. When the virus actually infects, it not have any much effect because already it is being defended, this papaya plants. So what happens when the virus enters into the plant? This plant was reduced to decrease the virulency of this virus, thus protecting themselves. Otherwise, if the RNA is broken down before it can be translated, then the message contained in the DNA will never lead to the production of protein. So this phenomenon is called as gene silencing. What they mean to tell is, so this is an RNA virus, we know that. So the RNA virus, what happens? It will get translated into to form a protein. Okay, so the translated, then the messages contained in the DNA will never lead to the production of the protein. So when you are destroying the RNA, where is the question of the formation of the protein? Okay, protein cannot be formed because there is no messenger RNA to send a message that what type of protein should be produced. So there will be no virulency. So this process is called as gene silencing. So these three methods were used to bring about the resistant variety of papaya against the papaya ring spot virus. Next, we shall see about the BT cotton. So we already know that this BT cotton, it is very common now. It is very well used commercially. These cotton plants are infected by the lepidopteran larvae, which destroys the cotton plants. Okay, so these are insects which belong to the family called as lepidopteran. So these particularly infect our cotton plant. That is, they completely destroy the cotton plant. So the BT toxin was isolated from Bacillus thuringiensis. BT stands for Bacillus thuringiensis. So they isolated this toxin from the Bacillus thuringiensis to see that whether could induce some resistance in the cotton plant against the lepidopteran. So what we mean by this BT toxin is, see, it is a delta endotoxin. This is how it appears to be. It is a crystalline proteins. They are classified according to the structure and have a specific nomenclature which are called as Cry1AC. This is the toxin. So from where did you get the toxin? I got it this toxin from a bacteria. And what is the name of that bacteria? It is the Bacillus thuringiensis. So what is my aim or goal of my experiment? My cotton plants are getting damaged by the Lepidopteran larvae. I want to destroy this larvae. So I'm using the toxin from the Bacillus thuringiensis. And which is the gene you're using? The gene I'm using is the Cry1AC. The cotton has been transformed with Cry1AC, which has a narrow spectrum. Narrow spectrum means this Cry1AC, this gene is particularly, it will target only the Lepidoptera and no other species. It is that is what it is called as narrow spectrum. So how it does so? This protein binds with the receptors in the insect gut, causing the pore which perforate the midgut and lead to the cell leakage and insect death. That is, you can see here, and suppose say this is my, of course, this is the cotton. Now what happens is this is the species Lepidopteran. Now what I'll do is, I mean the scientists they do is they'll isolate this gene Cry1AC. Okay, this is, as I told, it is an delta endotoxin gene Cry1AC. It is present in the Bacillus thuringiensis. These are transferred. Okay, here these will be. This is the toxin. These will be transferred to the agrobacterium. Which agrobacterium? Agrobacterium tumefacius. And then this agrobacterium, which is carrying the Cry1 gene, is transmitted into your cotton plant. So now what happens? Your cotton plant, okay, come here, your cotton plant is transformed. Why? Because this cotton plant is having that cry gene of Bacillus thuringiensis. So you isolated this cry gene from the Bacillus thuringiensis, a bacteria, and then what did you do it? You added this gene into the agrobacterium. We'll not go into the agrobacterium as you already know that. And you took this agrobacterium, modified agrobacterium, which is carrying that vector, okay, that is the cry gene it is carrying, and you infected into your cotton plant. And this cotton plant started to grow now. And then it flowered and there was cotton. Now what happens? These Lepidopteran, they come and they start feeding on the plant. They don't know that this is a modified plant. Okay. So now once when they start feeding, what happens? These transgenic cotton, when the insect feed on this plant, the Bt spores which are there in your plant enter the insects. The alkaline pH and the protease enzyme in the stomach of the insects, okay. So when they start feeding, what happens? The spores which are present, the cryspores or the genes which are present in the plant enter into the Lepidopteran. And here what is in the Lepidopteran, the condition? It is having an alkaline pH and protease enzyme. So the protease enzyme and the alkaline pH will become favorable for the cry genes. So the cry genes are actually, it is called as parasporal crystalline toxin. That is, it is not active. It is in the inactive form. So only when they enter into the insect, because of the insect's gut, which is having alkaline pH, and because it is having this protease enzyme, it will become active. The toxin will become active. Once the toxin will become active, what happens is it will create the pore in the guts of the insect. Okay. So the cry gene is a toxin form. Thus, it enters into the insect. It creates a pores in the gut of the insect. So now what happens? Now it will start to degrade there and it will start killing the organism. So The insects eat the Bt crystals and the spores which are present on your cotton plant. The toxin binds to the receptors in the gut and the insect stops eating. Once the toxin binds to the gut of the insect will stop eating. The crystal causes the gut wall to break. Okay, the gut wall of this insect will break down, allowing the spores and normal gut bacteria to enter into the body. All these bacteria will now enter, the toxins and other bacteria will enter into the insect. The insect dies as the spores and the gut bacteria proliferate in the body. This is how it is bringing about the death of the insect. So once again, I shall repeat this. So the scientists have isolated the cry gene. Actually, this cry gene is in the inactive form. They transfer this cry gene into the cotton plant using the agrobacterium Ti plasmid. Now this cotton plant is a transformed plant. It is carrying the transgenic gene of this toxin. So when the insect, your insect Lepidopteran, when it feeds on it, what happens is the toxin enters into the insect. The insect body is having a alkaline pH and it has a protease enzymes. So now what happens? The Bt spore enters the insects. The alkaline pH and the protease enters in the stomach of the insect and dissolves the protoxin. Okay, the protoxin means it is the inactive toxin. So the protoxin cry gene, it dissolves. What dissolves? The alkaline pH and the protease which are present in the insect, and they convert it into a toxic form, degrading the midgut by binding to the epithelial cells of the Lepidopteran. Thus, it causes the paralysis and the death of the insect. Soon as that toxin enters, no, the the insect will stop eating. And once it stops eating, this all process occurs in the insect, thus killing the insect. It ultimately the insect will be paralyzed and it causes the death of the insect. This is what we all spoke about it. Next, coming to your last one, herbicide. We spoke about this. We told that herbicide tolerant uh, plant are being generated. See, we add herbicide to a field to kill the weeds. But I told that when you're adding the herbicide to the field, the herbicide doesn't know that which is weed plant, which is crop plant. It'll think everything has same. Okay, so it will start destroying all the plants. So what we want to do is we want to see to that our plant of interest does not, it mustn't be killed by the herbicide. It should be tolerant to the herbicide. So one example is the Roundup. Roundup is the name of this herbicide which you spray to the field to kill the weed plants. So this herbicide contains what? Glyphosate. It is a broad spectrum herbicide. Broad spectrum means it is going to kill most of the herbicides. It is not like there are different type of weeds. Okay, it will be active against all the weeds. Which one? The glyphosate. So the active ingredient in the Roundup herbicide is your glyphosate. So the Roundup is your herbicide which is going to kill the weed, or it is going to kill the unwanted plant in your field. How it is going to kill? By this glyphosate. So it kills all the plant it comes in contact, because it doesn't know, it doesn't differentiate between the weed plant and the crop plant. So how it is bringing about the killing of the weed? It inhibits the key enzyme EPSP synthase in an amino acid pathway. So this glyphosate, what it will do is, how it is going to kill the plant is, it is going to interfere with this enzyme. called as EPSP synthase, which is an amino acid which is present in the amino acid pathway. This EPSP synthase, an enzyme involved in biosynthesis of aromatic amino acid, vitamins, and many secondary plant metabolites. So you see that EPSP synthase, this enzyme is very important. You can see that it is involved in the biosynthesis of aromatic amino acid, vitamins, and many secondary metabolites of the plant. Now, if your herbicide glyphosate is going to destroy this EPSP synthase, obviously my plant is going to die. It may be a herbaceous plant, unwanted plant, or a weed, or even my crop plant, because everyone is having this EPSP synthase. So the plant died because they lack the key enzyme amino acid. So a resistant EPSP synthase gene allows the crop to survive the spraying. So what you have to do as a scientist is you have to create a plant or generate a plant which is resistant to this glyphosate. That is, your EPSP synthase enzyme should be resistant to this glyphosate, or in turn it should be resistant to the Roundup. How is that done? We shall see now. See, this is the mechanism. Okay, so there is shikimic acid and phosphoenol pyruvate. You add glyphosate. Glyphosate is what? It is the active ingredient which is present in your Roundup. Okay, it is present in your Roundup. What is Roundup? It is an herbicide. What is its function? To kill the unwanted plant or the weed plant. But it can't differentiate the crop plant and the weed plant. It will kill all the plant. So once this glyphosate is sprayed, it is going to kill all the plants. How? By inactivating the EPSP synthase. When this EPSP synthase is inhibited, what happens? The aromatic amino acids are not produced. The particular vitamins are not produced. The secondary metabolites, what is required by the plants, are not produced. So what will happen? Without amino acid, your plant is going to die. This is the mechanism of how this herbicide is killing the plant, irrespective of your crop plant or the weed plant. Now, as a scientist, what we should do is we should see that we produce an EPSP synthase which can withstand this glyphosate. So the scientists have come up with an idea. So we had this shikimic acid and phosphoenol pyruvate, and this will get converted into 3-enol pyruvyl shikimic acid 5-phosphate EPSP. So what happens is, now suppose say I am spraying this Roundup to my field. What happens? We told that it is going to kill the plant. But here what we have done is we have modified a plant. How we have modified? See, that when we have spraying with this glyphosate, that is the Roundup, we have introduced a Streptomyces hygroscopicus bacterial EPSP synthase. That is, this Streptomyces is having a gene which codes for this EPSP synthase. Okay, and this gene we are going to put it into our crop plant. So once when we put it into the crop plant, what happens when you spray this Roundup? Roundup has no effect. The enzyme is resistant to herbicide. Okay, so this bacterial EPSP synthase is resistant to your Roundup or to your glyphosate. So even though you spray, my plant will not die because it is carrying the resistant gene from this Streptomyces. So what happened? All the reaction in the metabolic pathway will occur. Finally, aromatic amino acids and vitamins are produced and your plant will survive. So this is about the herbicide tolerant, particularly against this glyphosate. Hope you understood it. Okay. So the next one. eh, Hope you remember this glufosinate tolerant crops. Okay, even this is an herbicide. This glufosinate herbicide contains the active ingredient phosphonothrycin. Okay, there what we saw was Roundup. What was the active ingredient in the Roundup? It was glyphosate. Okay, so this is another herbicide, glufosinate herbicide that contains the active ingredient phosphonothrycin. So this phosphorothricin, even this herbicide, it kills all the plant. How it kills? It kills the plant by blocking the enzymes responsible for the nitrogen metabolism for detoxifying the ammonia. So what happens? The toxins build up and kill the plant. So this plant is also being modified. The crops modified to tolerant glufosinate contain a bacterial gene that produces the enzyme that detoxifies the phosphonothrycin. So what is happening here is you are inserting a gene from the bacteria which can detoxify this phosphonothrycin itself, and thus it can prevent the damage. So these are the two examples. This all you studied. Just now we spoke about this. Okay. So the glyphosate is a broad spectrum herbicide. This is a broad spectrum herbicide called as the Roundup. It is an active ingredient. I told you it is an active ingredient of Roundup herbicide, and it kills all the plant it comes in contact with by inhibiting the enzyme EPSP synthase. What is the function of EPSP synthase as you saw there? Okay, it inactivates the amino acids, the vitamins and all. So the plants die because they lack the key amino acid. So now what did we do here? We introduced the gene which can withstand the Roundup. And from where did we take this gene? We took this gene from the streptomyces. Okay. And then what happened? The EPSPS was now resistant to the Roundup. So when you spray the Roundup, nothing happens to this herbicide because it is already carrying the gene which is resistant to the Roundup. So the other one was the glufosinate tolerant, and the active ingredient is your phosphonothrycin. So how it detoxifies the phosphonothrycin you have seen. So this is how your